Yeah. Yeah. Okay. Good afternoon, dear attendants. My name is Dajo Smet. I'm very grateful for this opportunity to present my work on hormone balances in seedling development using a bioimaging method. So when seedlings germinate below ground, they face, of course, important challenges. The first challenge is the darkness. Plants need, of course, light to be able to produce their own energy. So seedlings need to become photosynthetically active as soon as possible. A second challenge is, of course, the overlying soil. So the soil is a major obstacle that hinders the seedlings in their quest for light. The seedling growth and development in complete darkness that starts from germination until the emergence from the soil and exposure to light is called scotomorphogenesis. So once seedlings are in the light, development changes from scotomorphogenesis to photomorphogenesis. Photo stands for activation of photosynthesis. So chlorophyll and anthocyanins are being produced, and true leaves start to develop. So why is this important to study? A lot of major crops in the world actually are characterized by a hypogeal germination, which means an underground germination. So we see a lot of very important crops here. So this emphasizes the importance to study this early phase of seedling development in complete darkness underground. In nature, successful germination and seedling growth are essential to establish and sustain species within plant communities. Also, a main agricultural goal is to obtain a uniform and rapid germination and seedling emergence after sowing. Losses at this early stage of seedling development are generally not recovered. So it's very important that this stage of development goes as good as possible. So this early stage of seedling development underground has a major impact on crop yield, which is also the case for seeds which germinate below ground, and so for seedling survival in nature itself. Hormones are very important regulators of plant growth and development. Interestingly, the regulation of seedling stem growth and development is different between light and darkness. So it's not because something is regulated in a certain way in light that it is also the case in darkness. So that's why it's very important, and this justifies why we study hormonal regulation of seedling development in darkness, so upon underground germination. But to protect the seedlings, so as I mentioned before, when seeds germinate below ground, they are of course covered with soil. So in order to protect themselves when they grow through the soil, seedlings of dicot plants develop a hook-like structure in complete darkness. And this hook-like structure is called the apical hook. You see a drawing of this one here. So it develops at the apical end of the hypocotyle. So here is the shoot apical meristem and the seedling leaves. And by this apical hook, they are directed away from the growth direction. So in this way, it protects these important parts when seedlings push through the soil. The development of this apical hook is a very dynamic process. So it proceeds in 3 distinct phases. We have a formation phase, so the hook forms until it's completely closed, and then this closed hook is maintained until seedlings emerge from the soil and become exposed to light, and then the hook starts opening again. And this process is regulated by a very complex hormonal crosstalk which involves multiple hormones, as you can see here. So this is a typical graph of which depicts the angle of Hooke curvature in function of time, and you see really always the same pattern here with the 3 distinct phases. So, of course, you can wonder, how can we study dark-grown seedlings? A key tool to do this is infrared imaging. So plants lack photoreceptors that are able to detect light with wavelengths longer than 850 nanometers. So this is where the infrared light is located. So plants are actually invisible for this infrared light. But using the right techniques, we can make it visible for us. So we use infrared converted cameras, and in this way we are able to see the plants in infrared light, but they grow as if they are in complete darkness. Our study on seedling morphogenesis after below-ground germination is focused on the model species Arabidopsis thaliana. So we developed an in-house dynamic infrared imaging system. You can see here a picture taken by an infrared converted camera of 2 plates on with medium, and each plate has 2 lines with small Arabidopsis seedlings. So using this dynamic infrared imaging technique, we can study seedling growth in complete darkness, and we can focus on the important processes, developmental processes during this morphogenesis. We can at the same time study seedling stem development, seedling root development, and apical hook development, and this at a high throughput and with a high quality. So here you can see a composite figure of one Arabidopsis seedling from seed on. So from seed on, germination, and then 144 hours later, this is the result of the growth of the seedling in complete darkness. So we can clearly see the seedling stem, seedling root, and the apical hook. So here it starts forming, here it's formed and maintained, and here it starts slowly opening. So this is all very nice visible for us. Of course, with these infrared images, we can also assemble videos. Yeah? These videos give a very nice overview of the growth of a seedling in complete darkness. So you can speed up a whole time frame of more than 200 hours in just a few seconds and really see what's going on the seedling level. So I'm now quickly going to show this short video. It's from germination on until more than 200 hours later, and it's with like 8 frames per second, so it's just in a few seconds. So you will clearly see the germination, the growing of the seedling, and you can also take a look at the apical hook, so the apical end of the seedling stem. There you will see clearly the hook forming, being maintained for several hours. I have to mention that also the hook also opens in complete darkness. It doesn't need light, but in nature is just in response to light. So germination, root growth, then you see hypocotyle growth and the development of the hook. Here the hook is maintained, and then it starts slowly opening. This is all in complete darkness on medium. So the opening phase is the more slow phase of the tree. Okay, so using this dynamic infrared imaging technique, we were able to add an important piece to the complex puzzle of hormonal regulation of apical hook development in Arabidopsis. So we focused on the plant hormones ethylene and breast-steroids, and we showed that there is an important interaction, a mutual interaction between these 2 hormones during the different phases of hook development. So okay, this was a bit my story. So I would like to take this opportunity also to thank some people I've worked with. So from the Institute of Science and Technology in Austria, I would like to thank the following people. And from our own Laboratory of Functional Plant Biology, I would like to thank my 2 promoters, Professor van der Straat, Professor van der Bussen, and colleague Yu Ming, which I also worked with. Thank you very much for your attention.
Time for a few questions.